Good evening, everyone. I am Dr. Satyabrata, MDDM clinical pharmacologist, and today we will discuss on innovative designs in clinical trials. And the agenda is precision medicine trials, master protocol trials, adaptive design trials, N of 1 trials. Coming to precision medicine trials. What is precision medicine? Precision medicine is an approach for treatment and prevention of disease considering genetic variability, environment, and lifestyle for each individual. It helps doctors or researchers to foresee the best treatment and prevention plan for a particular disease in specific groups of people. The aim of this strategy is to optimize patient outcomes by selecting appropriate treatment depending on unique molecular characteristics of the patient and malignancy and providing better safety profiles. Then coming to the types of precision medicine trials. There are lots of trials like longitudinal cohort studies, basket trials, umbrella trials, N of 1 trials, adaptive trials. Coming to master protocol. What is master protocol? FDA issued the draft guidance on master protocols, efficient clinical trial design strategies to expedite development of oncology drugs and biologics guidance for industry in September 2018. According to this guidance, a master protocol is defined as a protocol designed with multiple sub-studies which may have different objectives and involves coordinated efforts to evaluate one or more investigational drugs in one or more disease subtypes within the overall trial structure. A master protocol may be used to conduct the trials for initial dose finding phase or exploratory phase 2 or at confirmatory phase 3 to support a NDA, that is new drug application. Master protocol can be designed to evaluate different experimental drugs by parallel comparison to their respective controls or common control. Master protocol can be with a fixed or adaptive design. Different master protocols are basket trials, umbrella trials, and platform trials. Then coming to platform trials. What is platform trials? Multiple experimental treatment can be compared against a single control arm standard treatment, and without framing a whole new trial, a treatment arms can be added or dropped according to pre-specified criteria during a platform trial. Look at that image that shows the platform trial. One is a standard of care, intervention 1, then intervention 2, and after some time there is interim analysis and arm intervention 1 arm is dropped and new arm introduced, and that is intervention 3, and after that another interim analysis was done. At that time also new arm introduced. Then all the outcomes will be analyzed. Next is closed platform trials. Closed platform trial refers to no further treatments are allowed to be added other than those included at the beginning of the trial. And this is the most basic platform trial design. In this strategy, a fixed number of treatment arms in N samples is 5 and a shared control arm are used. Each arm also have fixed sample sizes allocated by equal randomization. Coming to open platform trials, open refers to adding new treatments to replace ineffective treatments during the trial. This strategy starts with a fixed number of active treatments n is equal to 5 and a control. Bayesian methods, a treatment response rate is greater than the control response rate, are applied to adaptively drop and add treatment arms at interim points until the success criterion is achieved by any one of the treatments, at which point the program ends. That means this trial continues in a perpetual nature until new experimental treatments are available to enter the trial. Coming to response adaptive randomization. A response adaptive randomization is a process by which participants are randomized to the study arms based on interim analysis of data to ensure maximal study efficiency and maximum chance of benefit of the participant from the best study intervention. This shows enrolling of the patient, then randomized to all treatment arm equally, then flexibility in allocating new patient, then treatment A, treatment B, treatment C, and treatment D, etc. Start of trial, then outcome analysis 1, follow up 1, outcome analysis 2, follow up 2, outcome analysis 3, follow up 3. Then from this, the flexibility in allocating new patient to the randomized to all treatment arm equally. Coming to multi-arm, multi-stage clinical trials, that is MAMS. MAMS design, multi-arm, multi-stage clinical trials design, evaluates and compares the effect of several experimental treatments, including combinations of those treatments against a common control group, standard treatment group, in a single randomized controlled trial. And start of recruitment, there is standard regimen, novel regimen 1, novel regimen 2, novel regimen 3, novel regimen 4. Then fast interim analysis, novel regimen 4 top due to toxicity, and novel regimen 2 the best performance, and secondary interim analysis was done after stage 2, and the novel regimen 1 stopped due to inferior performance, and novel regimen 3 stopped due to adverse reaction, but novel regimen 2 best performance. It will continue the end of recruitment, then after that stage 3 there is end of recruitment and at last final analysis done. And this is a complex trial design. Coming to basket trials, patients with same genetic mutation, irrespective of their cancer location, like lung, breast, colon, liver, or any other organ, are participated in basket trial. Characteristics: marker defined particular mutation cohorts, and non-randomized type of trial, and treatment is the primary aim of this trial. Flexible design, and it is flexible design, so continually open and close arms as the study progress. Basket trial, no control. So multiple diseases, target intervention, no randomization. Basket trial with control, multiple diseases, targeted intervention, common targeted interventions, then randomization, intervention and control, randomization to intervention and control arm. Then coming to umbrella trials. An umbrella trial includes patients with same basic cancer type like lung cancer undergone for molecular marker genetic mutation testing for identification of a wide range of potential targets. Characteristics: its aim is to facilitate screening and accumulation of patients. It involves a group of enrichment designs within the same protocol. It is a randomized type of trial. Flexible design, so continually add and drop arms or biomarker subgroups as the study progress. Umbrella trial, no controls, single DGs, but different intervention like intervention 1, intervention 2, intervention 3, multiple targeted interventions, and no randomization. Umbrella trial with controls, single DGs, multiple targeted interventions, intervention 1, intervention 2, intervention 3. After that, they are randomized, intervention 1 and control 1, and randomization on intervention 2, control 2, intervention 3, control 3. Advantages of basket and umbrella trials: may identify possible treatment options for small groups of cancer patients, even only 1% or 2% with a specific marker. For a rare mutation, oncologists may have an actionable treatment plan to suggest their patient. Multicentric trial with specific target mutation or marker. New potentially effective treatments can be identified faster than conventional design. Adaptive trial. The traditional clinical trial is a rigid channel of drug development process. Traditional clinical trials have 3 steps: designing the trial, to conduct the trial as per design, collecting and analyzing data according to a pre-specified plan. Adaptive design is defined as a clinical trial design that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the trial. Then coming to conventional vis-a-vis adaptive design in clinical trials. Adaptive design means a design that permits modifications of the trial procedures and oblique or statistical procedures of the trial after its beginning without compromising the validity and the integrity of a trial. So this is the schematic diagram of adaptive design. Design, conduct, analyze, then from conduct, interim analysis, reviewed, adapt, then conduct. Extra loop is there, and from design, conduct, analysis, this is the conventional design. Coming to adaptive randomization design, it is a randomization procedure that analyzes the patient's previous treatment allocation and its responses to determine the probability of future treatment allocation of new patients. Randomization and start trial: treatment 1, treatment 2, treatment 3. Then treatment 1 worst performance, dropping the inferior arm. Then treatment 2 best performance, increase allocation ratio, best performance, continue allocation, and final analysis. But treatment 3 moderate performance, constant allocation, moderate performance in secondary interim analysis, no further allocation, then final analysis. Standard treatment, constant allocation, final analysis. And in fast interim analysis, treatment 1 is showing worst performance and dropping the inferior. And final analysis of all the treatment 2 and treatment 3 and standard occurs. Coming to group sequential design. In this design, sample size of the trial is not set in advance and trial may be stopped before completion depending on the safety, efficacy, or both according to the results of interim analysis. An example trial using group sequential design. First sent from randomized, then interim 1 PFS progression free survival was seen, interim 2 OS overall survival was seen, then final OS overall survival was seen. Coming to sample size re-estimation design. In this design, sample size can be reassessed and adjusted during the study based on interim analysis to ensure adequate power of the study. Interim analysis at 187 events. Interim analysis efficacy favorable, utility promising zone, planned ends. This is the design. This is a very complex design. Then drop the losers design. In this design, inferior treatment arm groups are dropped and new treatment arms are added. This design is usually used in phase 2 clinical trial, especially during dose finding when there are uncertainties in dose level. General principles: drop the inferior, retain the control arm, may modify or add additional arm. Coming to adaptive dose finding design. This design is often used in early phase clinical development, phase 1, phase 2, to identify the minimum effective dose and or the maximum tolerable dose, or to determine the appropriate dose level by dose escalating for the next phase clinical trials. This is the diagram showing the adaptive dose finding design. Coming to biomarker adaptive design, design which identify most suitable target subpopulation with respect to particular treatment based on either clinical observation or known biomarker and evaluate the effectiveness of the treatment on that subpopulation in a statistical valid manner. Evaluation of biomarker: biomarker positive, biomarker negative of study, biomarker positive, randomize, new treatment, and standard treatment. Evaluate biomarker: biomarker positive, biomarker positive, randomize, new treatment, standard treatment, biomarker negative, randomize, new treatment, standard treatment. These are the design of biomarker adaptive design. Then adaptive treatment switching design. In this design, the investigator may switch a patient's initial allocated treatment to a different treatment depending on the efficacy or safety issues. Survival analysis is often challenging in this design, especially in oncology trial. If proportion of treatment switching is high, it could affect the study hypothesis and may lead to change in hypothesis. So adjustment of sample size is necessary to maintain the desired power of the study in this design, adaptive treatment switching design. Adaptive hypothesis design. In this design, modifications of hypothesis is done based on results from interim analysis. For example, switching from superiority to a non-inferiority hypothesis, or the switching between primary and secondary study endpoints. These modifications of hypothesis is mostly considered prior to database lock and oblique or data unblinding. The adjustment of sample size is necessary to maintain the desired power of the study. Coming to adaptive seamless phase 2/3 design. A seamless design joins two different trials like phase 2 and phase 3 into single trial, also known as operationally seamless. Primary objective of this design is to combine dose selection and confirmation phase into one trial. And in this case, randomization and start trial, then standard arm, regimen 1, regimen 2, regimen 3, regimen 4, then standard arm continued allocation, first interim analysis, second interim analysis, final interim analysis done. Regimen 1 after first interim analysis dropped due to poor or no response. Regimen 2 arm after second interim analysis withdraw due to inferior performance. Regimen 3 best performance, continued allocation till final interim analysis. Regimen 4 dropped due to toxicity and adverse reaction after first interim analysis. And from randomization to second interim analysis is the exploratory phase 2, and from second interim analysis to final interim analysis is confirmatory phase 3. Coming to multiple adaptive design, it is the combinations of any of the above adaptive design. Commonly used multiple adaptive designs are a combination of adaptive group sequential design, drop the losers design, and adaptive seamless trial design. Adaptive dose escalation design with adaptive randomization, and statistical analysis and inference of a multiple adaptation design is very difficult and often challenging. Then coming to N of 1 trials. N of 1 trials are multiple crossover trials, usually randomized and occasionally blinded, conducted in a single patient. N of 1 trials are a specific form of randomized or balanced designs characterized by periodic switching from active treatment to placebo or between active treatments, withdrawal reversal designs. An appropriate N of 1 trials designs include balanced sequence assignment, blinding, and systematic outcomes measurement. This is the diagram showing the N of 1 trials, trial design, baseline measurements, trial periods balanced or randomized, feedback for decision making, repeated outcome measurements, statistical analysis, and this is the design of N of 1 trial. It is very complicated design. Then coming to advantages of N of 1 trials. It determines optimal therapy for individual patients. It provides the strongest available evidence of treatment efficacy for the individual patient. Patients are more likely to have better adherence as well as compliance to treatment. Chronic disease management using N of 1 trials can improve patient management and save health costs. Identify cost effective treatment options. They may also prove therapeutic equivalence of drug. Coming to disadvantages of N of 1 trial, rapidly progressive conditions that may cause catastrophic outcomes. Asymptomatic disease may make outcomes assessment difficult unless a valid biomarker or biochemical test exists. Treatments with a very slow onset of action may lose interest of the patient as well as clinician. Treatments with prolonged carryover effects require longer washout period. Disease severity, if changed seasonally, may cause bias in assessment of treatment outcome. Multiple comorbidities with complex dose titration are not candidate N of 1 trials. This is the end of the my slide, and thank you very much for listening. Please subscribe and uh, like my videos. Thank you very much.